This is where we left off on the last teaching unit, and one of the leaving here in terms of the two major differences between prokaryotic, eukaryotic. Not only prokaryotics don't have membrane-bound areas where you have your DNA, but they vary vastly in size. The eukaryotic cells tend to be about 10 to 100 times larger than prokaryotic cells. And prokaryotic cells can be extremely small, nanometers, all the way up to something that is, as I said, about 10 to 100 times smaller than eukaryotic cells. As a cell increases in size, it also increases in its capacity for function. So many different functions can take place in eukaryotic cells that can't take place in prokaryotic cells. That's the good news. The bad news is as we move on, that volume to surface area creates some problems that we'll talk about in the next slide. Both of the cells have a cell membrane that maintains its environment. The internal membranes of the eukaryotic cells help to maintain an internal environment of that particular organelle, because that particular organelle has a function that's unique to the functioning of the cell itself. The plasma membrane is a very complex membrane in the eukaryotic cells particularly, and as a matter of fact we have a whole chapter where we'll talk about nothing but plasma membrane and the structure of plasma membrane. It's a biphospholipid layer, so you have phosphorus on the outside, which it tracks water, lipid on the inside, which repels water, and then embedded in that membrane you have various channels and proteins and items that help facilitate the metabolic functions of the cell. The double layer and the fact that it has a lipid component, has channels, has proteins, allows that membrane to bring some things in that are needed for the cell, and then also to get some things out, things like oxygen, nutrients, waste products out. The membrane surrounds cells, and then inside the cell you have the cytoplasm, which is a semi-fluid medium, meaning it's a little bit thicker than water, but water-like. You can think of it that way, to allow things to move around with ease inside the cell. In this image you see the design of the cell membrane, this phospholipid. So you have the phosphorus is a little purple dot, lipid is the yellow, longer, thinner piece. And you have two of those together, as well as you're seeing some proteins embedded in that membrane. One of them is showing quite an open channel where you can see that something can pass through. Now this open channel isn't just an open channel for anything to pass through, it's only certain things that can pass through. And then these carbohydrate side chains that it's showing, what they do is they allow things to actually be caught, so that then can be taken in a timely fashion. So they're kind of caught in that membrane outside. There are a few other things embedded in the membrane and other aspects of the plasma membrane. Again, we'll talk about that in chapter 7. A little bit about, or actually more than a little bit about this surface area versus volume. Surface area will increase while total volume remains constant when one goes from one unit to multiple units. And that's why most organs and organisms that have eukaryotic cells, the cell size doesn't change as the organism gets larger. So you are made up of billions and billions of cells that aren't any larger than the original cell, or actually I don't know this because I wasn't around when the original cell, they aren't really that much larger. Actually, to tell you the truth, they aren't that much larger than the original cell you started out with, because even though I wasn't there when your original cell started out, they actually can look at the original cell. So the sperm and the egg getting together to create a fertilized cell, your cells that you have in your body now are not particularly bigger than that cell at all. You just have many, many more of them. And that facilitates the metabolic processes that go on in your body. So looking here, what you have in the first block, total surface area, the formula is the sum of the surface areas, height, and width of all boxes, sides, and times the number of boxes. So that's total surface area. So in the first one, height and width, you have 1 unit, height and width. Surface area is going to be 6. That's in the first box that you see there. You move on to the second box, which is a single box, and its height and width is 5 times bigger, so it's 5. And so its total surface area, again, height times width of all boxes, sides, and number of boxes. And that then is going to, total surface area is going to be 150. So you only have 1 unit of it. It's much larger, but the total surface area actually, the ratio is much reduced. And in the third one, you have multiple units of the 1 by 1 size, and what you have is a surface area that then total volume surface area that is much more than the same size at where you just have 1, but as your number, your total volume remains constant. As the surface area increases, total volume remains constant. And so you can see this is illustrating how it's much more, if you want to keep the volume and surface area, so if you want the total surface area to remain constant, I guess I'm saying that the total volume to remain constant, you have to increase the number of cells as opposed to the size of cells. And you might be asking yourself, why do we care if the surface area and total volume are in correct proportion to each other? That's because all of these cells have to get things out of themselves and into themselves. And that becomes an optimal equation. It's critical. The surface area to volume ratio is critical to the functioning of that cell. So it becomes something that is important to look at and to have some understanding of, because it's the life of the cell there. Cells that have greater surface area relative to volume, small cells have greater surface area relative to volume. So again, it's just saying it the same way. It's saying it, I guess, in another way, that the smaller the cell remains, the more efficient it is, you might say. So it's much more efficient to have multiple units of the same thing. At the bottom row of numbers, you have the surface to volume ratio, and you can see in the block that's just 1, that's 1 inch by 1 inch, or 1 by 1 unit. We don't know if it's inches. And then on the third column, where you have multiple of 1 by 1 unit, you have the same surface to volume ratio. And then in the middle row of numbers, talking about total volume, how to calculate total volume is height, width, length times the number of boxes. And another compare and contrast. This is similar to the image that was in the beginning. We kind of talked about this already, that eukaryotic cells have internal membranes that partition the cell into organelles. And what we're going to do now is talk about those individual organelles. Plant and animal cells have most of the same organelles. Plant cells are going to have a cell membrane, plant and animal, cell membrane. Plant and animal are going to have nucleus, nucleolus, smooth endoplasmic reticulum, rough endoplasmic reticulum, ribosomes, vacuole, and mitochondria, Golgi apparatus. Plants, unique to plants, are chloroplasts, a rigid cell wall, and plasmosmatas are unique to, so those holes in the cell wall for things to get in and out are unique to plant cells. Unique to animal cells are centrosomes, lysosomes, and centrioles. First organelle that we're going to look at uniquely is the nucleus. It tends to be the largest organelle of the cell. It also tends to be sometimes the most prominent. Maybe that's because that's what we stain for. That makes it more prominent. But this is the area where DNA is stored. The genetic material for that species is stored. And DNA is what will direct the functioning of everything that goes on in the body. So the DNA of that cell directs the functioning of that cell, and then by expansion, those cells are getting together to form you. So it's directing the processes that go on in everything that you do. DNA and proteins form what's called the genetic material chromatin. So you have this word DNA and RNA. DNA is the portion that stays in the nucleus. RNA is the portion that can leave the nucleus. And if you put DNA, which is the nucleotides, together with proteins, which are amino acids, they form the genetic material we call chromatin. And chromatin is something that condenses into a term that we're probably familiar with finally, chromosomes. So when chromosomes are not condensed into individual units that we can see, chromatin condenses when the cell is getting ready to replicate. But when it isn't condensed, it's called chromatid. And the nucleus is surrounded by a membrane, and that membrane is unique. So it gets its own special name. It's called a nuclear envelope. An envelope because it does house the DNA, but it has special openings in it that allow the RNA to get out, plus also ribosomes to get out. It also allows, so it allows things to get out of itself. So the double membrane has pores, and it encloses the nucleus, separating it off from the cytoplasm, and there is a semi-fluid environment inside the nuclear envelope, which we call the nucleoplasm. Plasm is kind of our end word, which we call a suffix for fluid. So anytime you see that word plasm, particularly at the end of a word, you're thinking it's a fluid type environment you're talking about. The nucleolus, actually that word histones, that is it's a type of protein, but it's a term that's just used when you're talking about DNA, is histones. It's a specific type of protein. Nucleolus is located in the nucleus, and that is, so it's an area inside the nucleus, so the nucleolus, and that is specifically the site where RNA, particularly the ribosomal RNA, so little r RNA, and ribosomes are created. So nucleolus is where ribosomal RNA synthesis is created, plus these facilitators of protein production called ribosomes. We'll talk about those a little bit. And if you look at the image over there, you can see these things that we've talked about, and we will pick this up on the next teaching unit.